Our next speaker is Viviani Gomes, who has come all the way from Sao Paulo in Brazil. She's very welcome here to speak today, and she's going to talk on the influence of maternal antibodies on vaccine responses against viruses in Holstein calves.
Hello, good morning. First of all, I would like to say thank you for your attendance. I am going to show my research about influence of maternal antibodies on induction of vaccine immune response against respiratory viruses in Holstein calves. This research was developed at the University of São Paulo, located in the southeast of Brazil. The development of naive specific immune system progress in small steps from birth until 6 months of life. So, neonatal young calves depend on passive immunity transferred from cows as a primary basis for the protection. The level of maternal antibodies increase immediately after colostrum intake, following by a gradual decrease from birth up to 6 months of age. The duration of maternally derived immunity to respiratory viruses in calves is proportional to the concentration of antibodies ingested and observed. The decrease of maternally derived immunity is associated with the precocity of BRG. The rate of BRG is around 7.7 to 9.5 from second and until 30 months of life. It's the response for the treatment of around 21.9 animals. So, which will be necessary to vaccine young calves before the high period of susceptibility. However, we have a challenge here because the maternal antibodies have suppressive effect on the humoral immune response induced by vaccine antigen. So, the objective of this research was to evaluate the influence of maternal antibodies on vaccine immune response to viruses involved in BRG in dairy calves. For this, calves were assessed immediately after birth. First of all, the animals were evaluated by a clinical examination to select healthy calves. After, blood samples were harvested to perform virus neutralization and real-time PCR for BVGV to exclude fetal infection. We use colostrum donors. For them, we vaccinated the donors at 6, 7, and 11 months of life. They received booster twice a year using a commercial vaccine from Zoetis. The name is Cattle Master Gold. And every calves received the colostrum from the first milking in the volume equivalent of 10% of their weight in the first 6 hours after birth. 24 calves were distributed in 4 groups in accord with the vaccination protocol. For example, Group 1 received the first dose of vaccine at 14 days of life, Group 3 with 3 months, Group 2 with 3 months, and Group 3 with 6 months of age. And we had a control group non-vaccinated. The calves received the same vaccine that their colostrum donors by parenteral route. Blood samples. Blood samples were harvested at 2 days after colostrum intake to evaluate the passive immunity transfer. The vaccination protocol were evaluated at the day of vaccination, the day of the booster, at 30 days after the second dose. Results. for passive immunity transfer. The calves had high levels of maternal antibodies after colostrum intake, especially for BVGV and herpes virus. So let's start with the results from group 1, vaccinated at 14 days of life. All calves were seropositive for BVGV, herpes virus, PI3V from day 14 up to 74, probably due to colostrum intake. As you can see in this graph, the frequency of seropositive calves for BRSV had a slight variation. However, some calves already present some negative results in the virus neutralization on day 14. It can be justified by the low concentration of specific antibodies in the colostrum for BRSV. Okay, this slide shows the titers of respiratory viruses in the group 1. As you can see in the data, non-vaccinated group had high levels of maternal antibodies for herpes virus and PI3V at day 14. This fact also can be associated with the different concentrations of the specific antibodies in the colostrum. In general, in the group 1, the titers of specific antibodies go down from day 14 up to day 74 in both groups due to the decrease of maternal antibodies. So, the vaccination protocol of 14 days of life did not promote humoral immune response. So, let's look at the group 2 vaccinated at 90 days of life. We didn't find any difference for the frequency of seropositive animals between groups for BVGV and herpes virus. On the other hand, the frequency of positive results in the virus neutralization for PI3V and BRSV were higher in the non-vaccinated group. Unfortunately, this group had clinical signs for BRG around 90 days of life. Probably we detect BRG in the bacterial phase after virus infection. We also found high levels of antibodies for PI3V in the non-vaccinated group, because the same thing. Finally, you can see in the data for the group 3 vaccinated at 6 months of age. The non-vaccinated group had a slight variation during this study. On the other hand, the vaccinated group had an increase of seropositive animals after booster for all viroses. The titers of antibodies for BRSV, herpes virus, and PI3V also rise in the group 3. However, some calves did not respond for BVGV antigens from vaccine. It was our better protocol, but it is too late. In conclusion, the vaccination of dairy calves using parenteral route at 14 and 90 days of life did not induce humoral immune response. Calves only increased the titers of antibodies for respiratory viruses when they were vaccinated at 6 months of age. I would like to say thank you for São Paulo Research Foundation for financial support and my other partners, Zoetis, Biological Institute, and Colorado Farm. This research is a part of the master thesis from Bruno Toledo, and I could not let say thank you for my team, and thank you for your attention.
Thank you very much, Viviani, for a very interesting talk in a very important area that's often sort of neglected on the effect of vaccination and antibody responses. We've time for some questions from the audience, if there are any.
I would like to ask, in which classes had you measured the antibody response in that calves? In which immunoglobulin classes it was measured, and were any differences in the course of the immune response in that classes?
I didn't differentiate the class of immunoglobulin. I just did the virus neutralization for total immunoglobulin.
Did you evaluate the cellular immune response in these cows?
Okay. It was my first research with this kind of vaccine evaluation, and we tried to look the cellular immune response by phenotype, but we did it in the total blood, and there were a lot of factors that could be influencing my results. The calves vaccinated with 14 and 3 months of age had some response for the increase of gamma delta T cells, but I cannot confirm that this response is specific for the vaccine antigen, because there's other factors in the environment that could be influencing my result. Because that, I did not include this result in this presentation.
Hi, Mark Hilton from USA. You said you had some morbidity in the calves. Was there a difference between the vaccinated group and the non in morbidity?
Did you ask me about the difference between the morbidity in each groups?
Yes.
Okay. I didn't have any diet in this experiment because the control group was treated with antibiotics, and all of them survived. So it's my answer.
So morbidity, sickness.
Okay. Sorry.
Yeah, not mortality, sickness.
Okay, sorry. It's about 8% in the non-vaccinated group. Which were 4 calves and 5 in the control group non-vaccinated. It's 8%.
Yeah, Amelia Willems from the United States. Dr. Gomes, thank you for sharing that research. And I just want to follow up on the previous commenter and say that I hope you'll be able to look at cell-mediated immunity in the future, because we know that sometimes even when humoral immune responses aren't seen, when calves are vaccinated in the face of maternal antibody, we can identify cellular immune responses. So it'll be interesting to see what you see with the rest of that story.
Yes, I am doing a research now with an Argentine group that have the similar kind of vaccine that we have used in Brazil, because there's some difference. Like BVGV in vaccine in South America is allowed just inactivated virus. Then we need to look in others alternatives to improve this kind of the response. And I completely agree with Dr. Willems, because I think that this calf can be a cellular immune response, like another international experiment that we don't have these answers in South America yet.